Good morning, everybody, and welcome to the JKMRC Friday seminar series. On behalf of the University of Queensland and the Sustainable Minerals Institute, we would like to acknowledge the traditional owners and custodians of the land on which we meet today. We pay our respect to their ancestors and their descendants who continue cultural and spiritual connections with country, and we recognize their valuable contributions to Australia and global society. Today is my great pleasure to welcome Professor Gordon Southam. Professor Southam is an interdisciplinary researcher who has crossed traditional boundaries between biological and geological sciences. His research takes fundamental geomicrobiology and translates this into applied settings, primarily in minerals industry. Gordon's landmark contributions have been in understanding the fundamental roles that bacteria play in catalyzing the formation of economically significant geological and mineral phenomena, such as formation of placer gold, their role in mineral carbonation, bioleaching of metal sulphides, supergene systems, and bioremediation of mine sites. In this presentation, Gordon will highlight the role of bacteria in these geological systems. Today, his talk is going to be about the Geomicrobiology Laboratory at UQ. Please join me to welcome Gordon.
How's that microphone working okay? Thanks for coming out this morning. Just the first slide as the intro was going on, I know what's going through your mind, right? Do we have a big paw print or do we have a little rock hammer? And I can assure you that it's a big paw print, right? So we're going to start off with kind of how small bacteria are. We'll look at a bit of life in extreme environments and then get on to Kanga work in Brazil, some carbonation and gold if we have time there. So if we want to see bacteria, we have to get down to micron scale. And so the field of view here, we're looking at kind of 0.5 millimeter in size. And if we just zoom in another 30 times, we can start to see the bacteria on their scale. And if we want to understand their effects on geochemistry and how they dissolve minerals and form new minerals, we have to be looking at systems like this. In terms of the limits of life, there's bacteria and things will grow in ambient conditions. We often attribute the ability of bacteria to grow in extreme environments. And so here we've got a halophilic bacteria. They're growing in Basque Lakes in BC, and basically it's epsomite, it's Epsom salts. And so would you view this to be an extreme condition from a bacterial perspective versus an extreme condition from our perspective? And there's certainly microbial mats and biofilm growing within this environment. One of the weirdest things that happened, though, was we collected brine, we filter sterilized it, and when we grew bacteria on the brine, we had salt forming, whereas without the bacteria, there was no salt. And so we've got an evaporite mineral that actually is promoted, or its growth is promoted by bacterial metabolism. And it's probably just a change in water activity that these things do. Not really extreme. If you take these bacteria and you put them in fresh water, they'll explode, the osmotic effect, right? So this halophilic environment for these organisms is normal. Where we usually refer to extreme conditions is when these environments encounter people. And so here's Drayton's feet after running around the epsomite and sampling, and when he washed off the salt, his feet were looking like raisins because of the same osmotic effect that was coming out. If we go to Yellowstone National Park and Brimstone Basin, we can find sulfur oxidizing bacteria. The pH and the drainage around there's hydrogen sulfide hissing out of the ground, and it's pH 1. And so we have an extreme condition. If we were exposed to pH 1 sulfuric acid, we'd have a visor on and our leather bib and gloves and everything like this. In this environment, these bacteria, this is normal for them. I lost a really good pair of boots. My stitches just dissolved after running around the creek for a season here. We can go to high temperature, right, as another kind of extreme condition. And if you want to go to a really extreme environment, you start combining things. So if we can combine an acid environment with temperature, and Yellowstone gives us that too, then we're multiplying effects of extreme conditions on these organisms. A younger me, much younger me. To me, this is extreme from our perspective a little bit, but from the bacterial perspective is that the black that we're seeing there is iron sulfide. These are produced by obligately anaerobic sulfate-reducing bacteria that are growing at the Earth's surface. And so there has to be other organisms that are consuming oxygen above that film, because below it, even though we're exposed to oxygen out here, these organisms are still growing with in this environment. And that represents an extreme condition because the oxygen can kill these bacteria. And that's Monaco Heiberg Island. And a zoom of that it's just, I'd initially thought it was manganese, right? Because it's oxide, black, it's either carbon, manganese, or sulfides. And so sulfides were down my list, and we were corrected on this system. So let's shift into kind of biooxidation and another extreme environment, the Rio Tinto River that runs for hundreds of kilometers and goes right out into the Mediterranean. And so this is a challenging environment. It's pH 1 to 2, it's buffered with ferrous iron, and we get bacteria in these systems. And looking at this river, you can only imagine that bioleaching must be an easy thing, right? Because they're so successful in what they're doing in this environment. We can go to the Metcalfe pit in Morenci, where there's fractures, there's bacteria, and they're mining 400,000 kilos of copper per day with bioleaching. And so again, another example of bioleaching being an easy thing. We can zoom into that fracture, and you've got calcium right at the surface, and whenever it rains, that copper will dissolve and goes into their solvent extraction electrowinning process. And so an easy example of bioleaching there. They're even discussing the idea of stopping conventional mining and just pump fluids around, like copper, and they'll mine that way. And we've got our iron and sulfur oxidizing bacteria thriving in this kind of environment. If we go to Salobo, we can find an extreme condition on growth of these iron and sulfur oxidizing bacteria. So copper mining in Brazil. Zoom into the fractures here. We've got a bit of moisture. We've got organisms growing within this kind of weathering horizon in this environment. And when we look at these samples, we've got targets, we've got silicates, we've got sulfide, which was the main target. There was also magnetite and iron 2 silicates. And when we looked at the bacteria growing in this environment, we have our typical thiobacilli with our fibrous Schwertmanite gerocyte. We've got bacteria growing on the magnetite, and we've got bacteria growing on iron 2 silicates. So they'll take an electron from anything they possibly can in order to grow within the system. In terms of the magnetite, we produced a complete corrosion crust around the end. And just in this kind of higher magnification, you can see the bacteria that have been permineralized and trapped within that oxide boundary. For the silicates, it's iron 2 silicate. It's loaded with bacteria that are growing all through this system that are promoting weathering and potentially passivation of these mineral grains by the secondary mineral formation in this system. And so with all these examples, bacteria are extremely well adapted to kind of getting electrons from wherever they can. So if that's the case, why does stuff like this happen? Got a lab study, 4 students project. We've taken a consortium of iron and sulfur oxidizing bacteria. You put it on pyrite, you produce corrosion films, loads of cells. You put it on chalcopyrite, which is our big challenge, and you get minimal biofilm and minimal growth and minimal biooxidation. So what's the difference between these natural environments where it seems to be happening fairly easily, and then we try and control it, and it doesn't work nearly as well? So we've got kind of our work cut out for us here. Another example from Princess Creek, we're doing with Anita, that from an acid mine drainage perspective, the tailings has 7% pyrite. So it's like lots of acid generation. We've got a real problem here. The pyrite also, though, contains cobalt. It's like, oh, great, we've got an acid source, we've got cobalt, let's do a biooxidation. And we did it, and the cobalt that was in solution over the next month went from, trying to remember what it was, like 20 or 30 ppm to 0. Sulfur increased, acid increased, bioleaching was happening, pyrite was degrading, and the cobalt precipitated. presumably with the iron oxides. So we've got a challenge here. We thought this is an easy one, right? We can just bioleach the cobalt out of this Princess Creek tailings. When we collected samples from this environment, they were loaded with our regular iron and sulfur oxidizing bacteria. They're thriving in this environment. But when we looked at these samples with X-ray fluorescence microscopy, this is what we saw, that it is not a clean, simple leaching system, that we've got massive amounts of copper, iron, and zinc that are being mobilized. And if we zoom into the box there, you can see the black shapes. That's all the gangue minerals, right? That's your silicates, right? That are just kind of the background. But we've got diffuse copper through the whole system. We've got diffuse zinc through the whole system. And it's not leaching out as well as we would have hoped, given that there's 7% pyrite there. And so we still have things to learn and to work on here. Once you can put it in solution, there's a number of strategies in working with Denise Villagomez on recovery of metals with sulfate-reducing bacteria and bioreactor work, and that's what's growing in this system. She was interested in how can we separate nickel and cobalt that are commonly together. And what we saw with our nickel systems is that the bacteria would produce a brown hydrogenase, a nickel sulfur-centered enzyme that could perhaps separate these things a little bit. We've looked at gram-negative and gram-positive bacteria, and they do produce slightly different mineral phases. And over time, with a little bit of oxygen or under anaerobic conditions, we can make pyrite and framboidal pyrite and all those kinds of things with bacteria. Our real interest here, though, is in recovery of metal. So whether it's a nickel sulfide or a cobalt sulfide, biofilms will help in recover these metals in the system. And the real hope here is that we can start looking for proteins that are specific to these metals, that if they're important to physiology, bacteria should be producing proteins that can capture those metals, pull it into the cell, and use it for metabolism. And so getting into proteomics and the ability to produce chelators, in that case, you could produce affinity column, load it with that metal-specific or metal-binding protein and pass solutions through and then take the metal. And so could this be a formula for purification or processing of metal-containing solutions? Let's go to Brazil. Look at the iron systems that are there. So here's Brazilian rainforest. It's near Carajás, and this is just coming into land, and it is just green everywhere. It's lovely rainforest. When you get up onto the plateau, though, in the rainforest, on the top right, you can see the shape in the middle. That's the Discovery Plateau for the iron mines in Brazil. There's a little plaque at one end of the plateau, but you're still in the middle of the rainforest, and you're in scrubby, dry vegetation. That there's no soil there. It's growing on iron oxide, and what's happened here is that the iron oxide has actually limited the erosion that's happened on that flat surface, and the rest of the continent all around it has worn away, has dissolved away. Hundreds of meters of elevation of the continent has gone within the system. If you start digging into it and cutting open, you've got 5 to 10 meters of goethite. So this room, kind of thickness of rust, and you get down below and the silica is leached out. And the new mine, S11D, they don't have blasting, they don't have trucks. They're just shoveling it onto a conveyor and going onto the rail cars and sending it out. It's the richest ore kind of in the world. When you get at the surface and you start getting a closer look, it's an extremely hard surface, right? That we've got that 5 meters of goethite acting as a substrate for these organisms. If we zoom in on it, I describe it as an organic-looking rock, which makes geologists cringe a little bit because we don't really have organic rocks. And it looks like a stromatolite. We look like we have layers. And if you take a material here, this particular sample, I broke a chisel on. Just chisel, and I'm hitting it with a sledge to try and peel off some of this. And it is incredibly hard, and the corner of the chisel actually broke off trying to get this material out. And if you get a petrographic section and polish it, we've got layers and growth layers or growth bands within the system. And if we zoom into those growth bands, we find that it's full of fossils. And one of the original papers on the Kanga was a USGS scientist, and Dorr in 64 wrote this paper, and he said it's just too weird not to be biological in the article. And so we've got this whole thing here where we've got porosity and the porosity fills, and we produce this 5-meter Kanga blanket here. We can zoom in and have ion milled and polished samples, and if we can do nanosims on things like this and start looking at organic remnants and other things in the fossils. And so we've got carbon bonds, carbon nitrogen bonds, phosphorus associated with phospholipids. So we've got remnants of the biosphere that are trapped within the rocks, within this environment. So here's near Lake Violao. We're just kind of at the edge of the deposit. We've got organic carbon up here. It adds energy. You can have iron reduction. And when the iron-containing solutions flow out, they reoxidize, precipitate, and produce these cemented bands. And you can go from the ferrihydrite to just a dehydration reaction to goethite within this system. And so we were interested from a mine site remediation process is, can we reestablish Kanga? Can we grow Kanga? And Alan Levitt did a really interesting experiment in the laboratory. He had a bioreactor. He fed it with Kanga. He was feeding it with organics. And so you would take the iron 3 of the goethite into the iron 2. And then this is this kind of flow regime where it's just flowing over the bottom and there would be runoff. And when he finished the experiment, he was just like, how am I supposed to sample this? It's just, it looked delicate. It looked like he was going to destroy it. And he had to get out the hammer and chisel and lift it out. The whole piece was intact. It basically came out, started wailing on it with the hammer and chisel, and could break off chunks that he could use to grow it. So here's, you can see just kind of the gravelly texture off to the left here. So this is from that bioreactor that he knocked out with a chisel to get it into, ultimately to get it into plastic and make a petrographic section and have a look at this system here. And so here's where you grow in, here's the bits and pieces. And what we've literally got here is spot welds. And Alan figured out that it was maybe 1 or 2% of the iron had actually been dissolved and reprecipitated. You don't have to dissolve it all and precipitate it all to make that Kanga. You just have to produce a cement. And if we zoom into one of these spot welds, you can see a draping texture from wetting and drying. And if we go into this material here, it's all fossilized bacteria. So the cements in this environment are grown or are being grown by organisms. And so this represents an interesting way to stabilize or potentially glue and cement iron oxide wastes, so iron systems. Shift to mineral carbonation. We're Atlan, BC, northern British Columbia. They've got hydromagnesite deposits. They've got it's ocean floor ophiolite up in the Rockies there, and it's coniferous forest, so it's acid weathering, and it's flowing into this wetland where the hydromagnesite is growing. Greg Dipple brought us down to see the hydromagnesite, and within 30 seconds we're in the pond because it had biofilm and mine slimes and things that we're more interested in. So here's the pond itself. It's alkaline, high alkalinity, high magnesium. This is ultramafic rock that's weathering here. And if you look at the bacteria within this system, there's our cyanobacteria. In an alkaline system, they're going to take in bicarbonate, fix CO2, and pump out hydroxyls. So the pH goes up, and that's what's promoting the mineral carbonation reaction within this system. So here's our cyanos. There's heterotrophs associated with it. And when you feed it the solution from Atlan, we start producing hydromagnesite. And so we've got these platy minerals all in the exopolymer, and these are sheathed organisms where we're sequestering CO2. The biofilm that Janine grew here was about 3 times rainforest productivity, that if you feed cyanobacteria, you can produce huge amounts of biomass and promote the growth of these. The other thing that you'll find in kind of microbial mats and mine slimes is exopolymer. So it's kind of 95% water, 5% sugar, and it's like a gel. So if you picture all of these spiderweb textures here, it would be a gel environment in its hydrated state, not in the microscope. And we've got this idea of nucleation of minerals by exopolymer. And we'll just take a zoom into an early stage of this process. And you can see that the bulk of it is exopolymer, and we're just getting the minerals starting to grow. And so whether it's charge character or concentration effects is probably playing a role in the precipitation of these materials in the exopolymer. So keep the idea of exopolymer being critical for minerals. We'll come back to that in a few slides. We've done work on Heron Island, attracted to the beach rock, that it's like a pavement. It's a massive carbonate, and there's been arguments as to whether it's biotic or abiotic in its formation. And so if we get down to the high tide, here's high tide, you're looking at the dock in the distance. And if we go to low tide, this is what we've got. Where there's erosion, there's some breakage and things are coming apart. But you can see along here that there's different textures. And when you break beach rock open, or it breaks from storm events or things like this, it's self-healing. So it will fill in the gaps, re-cement it, glue it together, and stabilize beach rock within this system. Wherever you've got this, and you know, the abiotic explanations are wave action, degassing, and things like this, and effects of tides within the system. But on every beach rock surface, and certainly the younger materials, wherever you have water in this environment, you're going to have biofilm. And we can zoom into the biofilm here. We can see the oxygen that's being produced by photosynthesis. And in a seawater system where you're near saturated or partially saturated for kind of calcite or aragonite, the same reaction that takes in the bicarbonate, fixes CO2 and pumps out hydroxyls is going on. So the pH there is a little bit different. And if you look at the biofilm, we've got our same exopolymer and cyanobacteria and carbonate minerals that are growing that we've seen in other systems that we saw in Atlin, BC, and elsewhere. But if you take a piece of the beach rock and kind of polish it, we find aragonite needles. And the geologists are going to say abiotic. Geology wins, right? But if we take this material and treat it as a biologist would and chemically fix it and preserve the organics, then we get a slightly different texture where we've got aragonite needles in EPS. And if we zoom into that, the aragonite is forming an exopolymer. And so if exopolymer is precipitating the aragonite, did the biologists win? Is it a biological process? Or is it maybe a little bit of both of these systems here? We did some work with De Beers a few years ago, and this was the media release in 2017. They're going to pioneer a research program, right, to hopefully make carbon neutral mining a reality. This idea of could it be a possibility in 5 to 10 years? Okay, so that was the press release. We didn't actually get to the mine until June 2018 to start the work, so we were a year behind off the beginning, and we had a bigger challenge put to us because this is what De Beers put out, but this is what the press picked up. Okay, it's like we have carbon neutral mine in 5 years. And it's like, okay, so we're 4 years to go to try and do this. We got into, and you get into mine environments, whether you're in a subsurface mine and you have mine slimes that are on walls, or whether you're in a pit and you get water that's encountering in terms of water and kimberlite interactions, you can support this huge growth of biology, okay, that are growing on weathered byproducts within the system. Our photosynthetic organisms, right? So we've got the right geochemistry within these systems to try and sequester CO2 and to form mineral carbonates. And so we took the biofilms into a bunch of big kind of horse troughs. We grew them. We had microbial mat that was floating, others that were sinking. As they produce oxygen, they float, and then they kind of bubbles come off, and then they sink down again. And so we had to try and figure out how to inoculate this, because we're like, oh, can we scoop it with a pool filter or anything like this? And it was just, we're really concerned over what we were going to do. What we eventually did was we just took kimberlite from the mine and just sprinkled it into the horse troughs and the biofilm stuck to the kimberlite, and it was inoculated and sunk to the bottom, and we could just decant the water from the top and shovel it into the IBCs in the background there. And so here's a CRD control at 3 years. Nothing's happened. It's just, it's there, nothing's going on. In the cases where we inoculated with the biology, by 1 year we had plants growing. We didn't plant them. That's just stuff that's blown in. The same thing would have been in the control. Like there would have been seeds blowing into this. The plants could not get a hold within this environment. In the one where we accelerated weathering with microbiology, that it provided an environment to trick the plants, or the plants thought it was a soil. Well, we'll just germinate and we'll grow. And when plants grow, the rhizosphere becomes a really active environment. And within kind of our 2 to 3 years, all of the kimberlite was cemented within the system. And so we've got weathering of ultramafic rock. We've got kimberlite that's being glued together like we saw with the iron system. So we've got an iron cement. In this case, we've got a calcium cement or a carbonate cement, and this system with the main image with X-ray fluorescence microscopy is, remember, bacteria are micron scale. We're producing kind of millimeter scale carbonates within the system. And based on just a kind of a roughly 2% mineral carbonation rate, that within 3 years for this sample was offsetting about 20% of mine emissions. So that carbon neutral mine is not a geologic time phenomena. That is something that maybe we should be taking a little more seriously in the system. The most recent work we're doing in this was a IODP, International Ocean Drilling Program project to the Atlantis Massif. So it's middle of the Atlantic, spreading center, and the group managed to drill. The previous record was 201 meters of peridotite, and we went down 1.265 kilometers into some gabbro-hosted material, but mostly Harzburgite and Dunite, the olivine end members within this system. So here was, I was just thrilled to get this sample. The microbiologists, the geologists first looked at it and went, it's ruined. It's been weathered, there's been seawater circulation, it's not interested in it. The microbiologists were after what's the hottest, deepest, saltiest, extreme environment you can see, and they're like, we're only with 6R, we're only 30 meters below the ocean surface. And they're like, seawater circulation, we're not interested. I'm like, mineral carbonation, folks. It's like really interested in this system. So here it is, an RGB map where we've got iron, calcium, and nickel for the RGB. And so we've got carbonates that are forming here. We've got a little bit of iron, and so there's questions on serpentinization, and then we've got this kind of this overlay or this background of nickel. And it's actually best. I know we usually produce RGB so we can see these elements associations, but the element associations here are actually discrete. And so within this system, it's actually better to break it down to metal by metal to see what we've got here. And so if we start with nickel, you've got a nickel background in the matrix here. We've got large fractures with kind of the dark horizons there, and we've got very light or thin or shallow fractures occurring within the nickel-bearing material. And so if we shift from nickel to iron, all of the fine fractures within the nickel-rich material are actually enriched with iron. And in serpentinization, you're going to produce hydrogen, which is key for life in these systems. But we've got magnetite here corresponding to the part where the nickel is starting to weather and leach away. I was hoping we would be able to track nickel, but it seems to be going into solution and completely washing out of the other systems to turn up somewhere else. For the larger fractures that are here, that's where we get our calcium signal. So we shift into that. And it looks kind of like Loki's timeline with the spirals and everything that's there. But this is our mineral carbonation process that's going on. That's breaking open the peridotite. And when we went looking for DNA within the sample, we found it only in the carbonate. Okay, so we've got biosphere that's going on here, and promoting mineral carbonation within this, this system. Forget science papers and things like this. This is my new measure of success in science, that you can appear in a cartoon. And so while we were there on board the ship, it's like, oh, you know, Terminus Lagoon, log into the computer. So we have about 8 of these cartoons. Every day there would be a new one telling our story of drilling the Atlantis Massif. Let's shift to gold. It's the geologists that have told the story, gold nuggets grow. It's this idea that they increase in size and will accrete together. So these are samples a colleague gave us to look at. The hypogene material, the black is silicates. We got various mineral sulfides there, and the gold particles are on the order of 10 to 20 microns. If you go 50, 60, 70 kilometers downstream, the gold grains are a couple of hundred microns. And you can see within this that there's clearly discrete bits and pieces that have been cemented and glued together within this system. And so we were interested in looking at this process. And so one of the first experiments we did was looking at weathering of these materials, adding in our iron and sulfur bacteria, the same ones from our iron oxidizers, and see what we're going to get within this environment. And this is the kind of thing that we produced, that within this, we got loads of bacterial biofilm. We've got holes and nooks and crannies that are in there. We looked to the water to see if gold had been dissolved, and we didn't find any gold. And we went to the sediments to look for gold grains that would have been released, and we didn't find any gold there either. And Maggie Lenke, who'd done just a ton of characterization work, was just like, I'm not doing this again. Let's get a student. So Jeremiah Schuster came along, and he did the experiment, and he went looking for the gold in the solution or in the minerals that were left over. Couldn't find the gold. So we did it again. Couldn't find the gold. Reprepping, polishing rocks, everything to do at this step. On the third try, he put the whole experiment, rather than trying to separate out the fluids from the solids, he poured everything into a Petri dish. And as he was focusing down, there was a little flash. He's like, what's that? Came back up, found the little gold grains floating on the surface of the water. Now, when we grow sulfur, sulfur tends to float on water. It only has a density of 2, but it's really hydrophobic. And so the only thing that I can think of is when gold is initially released by weathering, it's extremely hydrophobic, and that provides the potential to be transported rapidly and far away from where it was leached and weathered in the first place. And so these are the gold grains. This is a gold grain that was floating on the surface, on the meniscus of the water in this gold weathering experiment. And we've been interested in gold weathering, right? Well, gold's cool, but you know, it's inert, it doesn't corrode. Yet, when you look at gold grains, and so here's some gold from Kilkeevan, panned out secondary electron image, so we've got kind of 3D, you can get all the scrapes and knocks and things that happen to gold grains that would, you know, it's like gold is very soft, and you can imagine if you're dragging it down a stream bed, you think it should, you know, kind of this idea of weathering and breaking down. So if you weather gold, how can you then grow gold grains? It's opposite. If we look at this sample here with backscatter electron, so high atomic mass is bright, low atomic mass is dark, and we've got within this, we got nooks and crannies and these idea of corrosion pits, so gold dissolving. But if we zoom into these dark horizons here, we also see gold precipitating. Now in this system, gold and silver dissolve, the silver is lost. So that's a different signal than what's going on with gold. A lot of the gold though reprecipitates, and you've got within the circle here, crystalline octahedral gold. You've got the 6 sides of the hexagon plus the top and bottom. So we've got crystals that are growing and it's pure gold. So the primary material will have, you know, 5 or 10 or 20% silver or something that's in there. And we've got colloids and crystals that are growing within the system that are pure, that can only be there if gold is dissolved and is reprecipitated in the system. We're interested in octahedral gold because we can make octahedral gold in the laboratory with our cyanobacteria and gold chloride and let it react. And we're getting colloids to crystals. And so we got this link to a biological or biological processes within this system. Carla Zammit did some really interesting work where they're looking at protein responses to gold, and 59 of these over 1,000 proteins that were associated with Cupria vitis is a classic metal-resistant bacterium that's out there. It's got resistance mechanisms to almost the periodic table, and so in this one there was 59 proteins that were specific to gold, and so if you could go through and sample soil and find these same proteins, would that then tell you that there's gold within the system or there's exposure of gold? And this idea of proteins and targets for not only chelation but for exploration aren't limited to gold or strange metals. That this is sites from Mount Isa region. We've got Mary Kathleen in here. We've got Ernest Henry. We've got Mount Isa, and these are genes that will produce something that interacts with a metal. And so the top of the scale here is 2,500. And so we've got in each of the sites hundreds of molecular targets that could be used in exploration. So just to pick on the geologists, you can do your soil survey and you can do weak leaches or harsh leaches, but you're only going to get maybe 5 or 6 different kind of metal signature processes. We've got responding to copper, arsenic, iron, chromium, selenium, tellurium, silver, zinc. It's the periodic table that's there. And so that there's hundreds of genes that could be used in a survey to complement the geochemistry and maybe say that there's a metal-specific process going on in this system. Gold is supposed to be toxic. Yet we were able to, it was just a kind of a sand column feeding the bugs that are in here. We could produce gold biofilms. And this particular experiment was right at the point where we were starting to get those new field emission microscopes operating at low KeV. And so we went from imaging something like this to being able to image the same biofilm like this. And it's revolutionized our ability to see bacteria and to see their effect here. That you've got the individual bacteria, the sulfovibrio that are here. We've got the hexagon of hexagonal gold or octahedral gold that's starting to grow. And that we've got these, we thought initially exopolymer, but increasingly we're thinking there's a huge revolution going on in microbial ecology related to nanowires. And it's the ability of organisms to have cytochrome chains, and it's you can oxidize a substrate, and you can use this wire to plug into a mineral and dump electrons. So you can promote iron reduction or manganese reduction. And in this case here, we're promoting our gold reduction. So the gold that we thought was going to kill our biofilm actually is precipitating away from the cells, and they're not being killed. When you do experiments with copper or iron or arsenic or things like this, you finish the experiment, you dispose of it. You do an experiment with gold, and I don't know if you've done work with gold, but you finish the experiments and it's like, let's put it in the cupboard. It's gold, right? We're not going to throw it away. So we've had experiments. This one here was iron oxides that had precipitated colloids, our cyanobacteria making our octahedra. We had our gold film and our biofilm that was growing on the surface. And so we had a cupboard full of these experiments and little columns and sitting in the grad club one afternoon, we thought, we've got to do something. What are we going to do we've got to clean out the cupboard. That was more of the issue that we needed the space. And so Jeremiah came in on Monday with a tumbler. So here's the microbiologist view of a river, right? And so we took all of our gold waste, we chucked it in the tumbler, we turned it on, and we left it for a month. And so we approached the month and it's like, how far has this thing traveled? Okay, here's our circumference, here's our speed, here's our rate, 80 kilometers. We went 80 kilometers in, just like the South American gold system that we looked at. And so when we opened this system after its 80-kilometer journey in 30 days, this is what we had. And it wasn't one, it was 10 5-millimeter, 1-millimeter, 2-millimeter size grains that grew within that tumbler by the action of bacteria. And we don't entirely understand what was going on, but just an incredible result, because we started with those colloids and little crystals that were produced by the bacteria. And we've got our tumbling effects and scratches and rounding and all the things that you'd expect in a natural system where gold grains grow. Stop there.
Perhaps we should go back to show the picture of the gold.
Of the gold.
All right, we have time for questions. Okay, one on the back. Yeah, for the online audience.
Thank you, Gordon, for the presentation. You show a lot of extreme environments. My is more a curiosity than a question. Black smokers in the mid-ocean reach, they are really extant. The environment is pristine. There could be now or in the future any possibility to study those kind of microbiological community. Maybe
there's definitely a community on the outside of the smokers, not within it. You're what, 350 or more degrees C, and so the temperature record for life is 122 degrees C. So it's at basically at autoclave temperatures under pressure. That anything that's degassing will be toxic, right? Like, you know, can you boil your water when you camp? So you can find them in the case of the IODP expedition. That was near the Atlantis Massif. And so you got massive carbonate spires on the ocean floor. That's a warm vent, kind of 90 degrees C, and so loaded with life within that system. The other one that's interesting is iron manganese nodules, right? Because there's iron manganese oxidizing bacteria. And so you find when you look at those nodules, they're full of fossils and remnant organisms that are there. I just got some nodules from colleagues in China, and it's on the edge of the Marianas Trench. So you got manganese nodules from 6,000 meters, right? I've got they gave me a little present that has Marianas or has Marianas Trench water and sediment in a sample. So that's an 11K sample that's there. So you're going to find bugs in all of those environments, and certainly wherever there's mineral substrates. That's just adding food. Right, the weirdest things in those environments are those tube worms, right? That they described it as a worm, but there's no mouth or digestive tract or stomach or anus or just anything that you'd expect in a digestive system. 50% of the biomass is bacteria. So you got this worm-like structure that has a red hemoglobin that can bind H2S and oxygen and give it to the bacteria that oxidize it, fix CO2, and share with the other organism. So anywhere there's substrates for life, there'll be bugs.
Thank you. I will just follow my the question that Francesco was doing. Thank you for the presentation. It was fantastic. And I have a question regarding more like the metallurgical and more like the economical point of view. Where do you think is the gap, or what do you think is the bigger gap now for the industry to apply these bioleaching solutions in great scale? And I know some also like developments some mining companies are doing as well. I spent some time in tech in Canada, in the British Columbia last year, and they have their own laboratory, like bioleaching laboratory as well. So they have biologists working inside the mining,
It's crazy town,
the
right?
mineralogical like hub that they have there is amazing. And yeah, I was working with the mineralogical team, but they were like these scientists, like all like doing stuff and trying to upgrade this like in a bigger scale, right? Yeah.
So I think there's obviously opportunity there. what is it? Is it like 15 or 20% of world gold or world copper production is with bioleaching? So it's there. It's being established in I guess places all over the world. I was just Nick Cook sent me some papers to read on samples from Finland or a mine in Finland where they're low-grade materials, but it's just the deposit is just truly massive, right? In the case of Morenci, right, the hypogene ore there is about 0.2, 0.25%, and the supergene is 0.43, and that takes it from not economic to economic. And so, but when we've looked at our systems that we thought were easy, they weren't easy. So Rick's complex ore bodies and what are the gangue minerals and how are they affecting stuff that's in there that Princess Creek, it's like we've got copper, loads of copper and zinc within the system, and it's like, why isn't it leaching? Why isn't it coming out? And so we have to sort that, right? And that'll improve recoveries. Yeah.
Thanks, Gordon. That was fascinating. I'm struggling to understand what's going on where you go from the colloids and then the tumbling in the river or the tumbler, and you end up with the aggregated nugget. Can you explain what's going on there?
That's what I said in the talk, is we don't know what's going on.
Really?
Right? So
Okay.
we certainly see it within the cultures that that the gold uptake basically, if you take a bacterial culture, you spike in, so there's a range of complexing agents from gold chloride to gold thiosulfate and stuff that's there. If you spike in a gold chloride solution, put it in a centrifuge, pellet it down, take the supernatant and put it into, at that time it was graphite furnace AA in the day, and zero gold. The gold had been picked up by the bacteria, and within probably a couple hours, it would go from kind of a gold-colored pellet to one that was pink or red, and red equals colloidal gold. And so we made colloidal gold as an EM tracer in grad school. It's like, oh, I got a pink solution. You look at the microscope, we've got colloids. If you look at that sample, you just allow time. So the cells have been killed, but all of the organics are in there, right? And so we've now got an abiotic, organic-rich material where the crystals are growing, and they grow over time. And then you'd start to see crystals aggregating together as they start forming gold grains. So, the cell guts is like every organic that you could imagine is going to be inside those cells. And I told my supervisor, I'm like, the gold went into the bacteria. And his whole research career was based on metals interact with bacterial cell surfaces. And he said, impossible. He said, I've done it. The gold goes into the cells immediately. And he went and pulled some files, and he had an experiment with gold wall, or with cell walls, plus gold, and it precipitated on the surface. And he said, what do you think of that? I said, it's fine, but if the other organics are there, this other reaction isn't happening. And so, there's thousands of different organics inside a bacterium. I didn't share it today. There was a gold specific protein that was produced that we worked with colleagues, and it would initially bind the gold. And I said, are you getting any minerals from it? He said, no, it's a pure soluble complex. And once we looked at it at the microscope, we saw that there was small colloids and crystals growing in that system too. But there are chelators for gold, and so the hope is that they don't precipitate and that you can recover gold. But we don't know, right? It was just throw it in a tumbler. Let's see what happens, right? Yeah.
One from online.
So there's a question from BR Palumbi. Great presentation. How were you able to separate genomic DNA in the calcium, nickel, iron mineral, specifically from each element fraction?
Yeah, so in the for the IODP sample, So we've done work. You can extract DNA. You just treat it like a soil. So the stuff we had a visiting scientist from China and looking at mineral carbonation, and we give her samples of the beach rock, right, to look at the surface microbial community versus microbial communities inside. That it's easy to precipitate carbonate at the surface. It's harder to understand how it's forming at depth. And so that was the comparison that we were going in. And if you just do like a power soil DNA extraction, that'll get it out. For the IODP samples, where we've got these fibers and networks, that crosshatch pattern, right, of carbonates that are in there, we had Sam with a little Dremel tool, sterilize the tool, and then go in and just dig out the veins. And so targeted DNA extraction there, and that's worked as well. It gets much harder with the Kangas samples, that especially where you've got fossils where the cell guts have been lost, that if you're losing that, then you're losing the DNA. So we got fossil remnants of things, we've got living organisms we can isolate and culture, but not all of it will have DNA that you can get out.
There's another one from an anonymous attendee. Thank you for your great talk. From your experience, how do you think bacterial populations establish and maintain themselves in waste rock piles during sulfide self-heating? Do you think they survive in 100 degrees Celsius and higher temperature range?
So in mine tailing environments, or
Waste rock piles.
So the whole exothermic heating thing that goes on. So there's been similar studies done in agriculture where you can have grain storage, and if you have water, you haven't dried the grain properly, that it supports metabolism, and you can have heating stacks and even explosions and things go on. In the case of waste rocks, you can increase temperature and that's the thing, if you have if you look at hydrothermal environments, you can find organisms that are, hyperthermophiles, right? That they thrive under high temperature conditions, and that environment has selected for those organisms. If you have a waste rock pile that's heating and building up temperature over a while, then you will select for bacteria that can withstand those higher temperature conditions. And so the proportion of the population will shift towards a high temperature organisms. And so the issue at surface is that you can't go above 100 without degassing. In the case of Yellowstone, with the elevation there, about 90 degrees C was boiling, right? So that was the temperature limit for life. So if you have an environment, the bugs they're probably there, or they'll be enriched, and then they'll grow within that system.
Okay, that was a really fascinating talk. It was like there's so many questions. It was really great. With the gold nugget, like I'm just baffled with that. But have you tried throwing something else in your tumbler, like something that's sort of equally as malleable as gold? Like a lead thing or something, to see if there's some mechanical input into the nuggety formation.
The lead would be tricky. That it's gonna precipitate, right? That getting from a lead 2 to a lead 0 would be a much more difficult thing. Lead is it's with electron microscopy we use lead citrate as a stain.
Yeah.
Right?
So
the
that was
citrate
just
helps
sort
stabilize
of,
it, but it's to prevent it. And we have, we'll have little sodium hydroxide pellet to grab the CO2 so the lead carbonate doesn't form. But we tend to get, it's difficult to put lead into solution, like a plumbogericite will precipitate, your lead carbonate will precipitate. And then you've got lead 2. And so you could, those salts could erode, but it's not going to behave the same as gold will. And we've looked at, kind of colloids and crystals formed with silver, with platinum, with palladium, that they're all, it never seems as active as the gold in terms of the crystals and things, that we seem to get colloids, and that's where it stalls.
Yeah, okay. And just as an aside, the precipitation on Heron Island, that sort of cementing and stuff.
Yeah.
Is there any work done looking at systems like that to with the view of looking at like coastal erosion?
I don't know if there are. I think it's helping protect Heron Island, and probably all Great Barrier Reef Islands have these cements. So that self-healing aspect of the beach rock, we took that experiment to an nth degree. We grabbed a sample of beach rock, and we chiseled off that surface biosphere, and we hit it with a hammer, and we took it back to sand, and then we took the lower material and bashed it with a hammer and took it back to sand, threw it in a little, again, one of our little kind of shoebox bioreactors, and Janine had 2 tides a day, right? In and out, in and out, in and out, over a 2-month period. And the one thing that she did that was really cool, because we wanted to say that microbialites are responsible for cementing beach rock. And so she added excess strontium into the seawater. It naturally has 8 ppm. You can bump it to 40 without saturating. And so we were able to trace the strontium to show us what the new microbialites were that had grown within the last 2 months. And they were all full of fossils. So the cement was biogenic within that system.
Sorry, Tom, do you want to? Okay, I'll be quick. Thanks, Gordon. So just a similar question to the previous question about the self-healing. A common problem in a lot of iron mines in Western Australia is the pit walls.
Yes.
And the AMD coming from the pit wall. It's an operational problem. It's not a closure or rehab problem. It's an operation, working in the pit. And so they use all kinds of things, shotcrete and, organic sealants and other things. But, it's not practical a lot of times. And I was just thinking, is there a possibility? I know you showed the example from Brazil, but in Brazil, you have those soft silicates like garnet, for example, but these deposits are shale hosted, and they're in shale, so the sulfide pyrite is in shale. And I was just wondering, if you wanted to design an experiment, I'm thinking about the proposal, thinking how can we, create a self-healing environment that, that prevent that.
So it's trickier in an AMD system, right? That you can have, you can have the iron oxides. My first mine tailing I went on, right? It was be really careful. I wanted to get some samples, and I was, advised to be really careful that there's hardpan through here. And I'm like, oh, how hard can hardpan be? I think I almost broke my wrists, right? Just hitting it with a hammer, and it only went in a few centimeters. And so you've got iron oxides, right? That can serve. Gypsum can help a little bit. You can have silica dissolving because of the acid, so like a silcrete kind of form. It's interesting. You look at mine tailings, and we looked at one, and it was 8 months old, and we had oxidation down to maybe 10 or 20 centimeters. You go back a couple of years later, and you've got kind of 30 centimeters. You go to a mine tailing that's 50 years old, and it's only eroded down maybe half a meter. And so what I think happens is that you have active oxidation that accelerates until you reach a point where the oxygen is consumed, and then it just stalls. And so it's going to stay and cook a long way. And then slowly over time, the whole thing will start moving down. But that's geologic time, right? That's probably hundreds, if not thousands of years for a mine tailing to weather that much. If you can capture that oxide horizon, and the thing is, even though you produce this and you're making hardpan and all these things, acid mine drainage doesn't stop. So it's not, it can give some physical stability. It can probably affect erosion a little bit. But in an AMD system, if the oxygen is not being consumed there, it will flow down to the point where it will be consumed by the bacteria. So it's versus the Kanga or an iron mine system that's not sulfidic and not an AMD system, that in that case, it's based on redox, right? That you've got the goethite there. Iron reducing bacteria can respire iron. They make iron too, and it will flow and move until it encounters oxygen, right? And so, where we had those outflows and those massive iron oxide films, that was coming from a reducing environment underground. And we saw some areas there that almost looked like a cross section of a pipe with layers, you know, that they're constricting, and so they affect the hydrology. But if it just pushes water out to another oxygen-rich environment, then it will precipitate and stabilize. So much greater chance of success in an iron mine than something that's sulfidic. Yeah, so we envisioned, right, and this was just right at the start of COVID, right, that it was like this idea that we would have huge, because we had cubic meter bioreactors that we were growing with our Kanga experiments. I've got a sample of iron oxide on my table that is just completely cemented together from that biofilm. But imagine setting up a biofilm here and just pump the solution over and let it flow down that face that we had with the Kanga. Only 1 or 2% of the iron was cycled to make those little spot welds or crossfits. So you don't have to dissolve, you know, produce a complete iron sheet. I was interested in, you know, you look at concrete and you've got gravel and sand and then your cement. And so with iron formation, you could have gravel-sized, you know, kind of waste. You could have sand-sized waste, and you would formulate it like you would be thinking of a concrete. And then the cement would be that iron oxide in the system. And so could you produce like the Kanga or like the beach rock, a roadway based on iron? And I think you can. But in the case of slopes, can you cement the material, that's the soft material that's there, or the loose material that's there, onto the bulk structure? And that would be a neat experiment. Yeah.
Well, please join me again to thank Gordon. I do appreciate there's still more questions, but the conversation doesn't have to start here. Stop here, so we can still have conversations afterwards. Next week, we have Professor Ross Macari from the School of Mechanical and Mining Engineering talking about automation in mining. So please join us next week. Thank you.